Hello everyone. Hi, my name is Himanshi and I am a PMRF PhD candidate at IIT Kanpur in BSB department. So today we'll be discussing about the previous year assignment questions for week 7, and I guess we can wait for 5 minutes so that people can join in, and then we can start with our weekly discussions. Okay, good. Um, I guess we can start with our discussion, and in the meantime, people can join in. And even if you're not able to join in today for any particular reason, you can always go back to the YouTube channel and look for the discussions. Okay. So, and I hope that you guys have completed the week 7 lectures from the portal. There were 5 lectures and they were predominantly for a neuronal system, as we are discussing neuronal system to a pretty extent, so that you get a holistic overview of the neuronal system, at least one system of in our animal physio. And we'll get all other systems also in the coming weeks. So hope you all of you are enjoying the course till now. And congratulations that you have covered half of the course, because it's a 12-week course, and you guys have been very, very, very patient, and hope that you have uh, applied for the certification and you guys are preparing well for it. You don't need to put any extra time, as I always say, and I'm repeating myself again and again. So you just need to follow the course lectures, follow the live sessions, ask questions, ask doubt from the TAs of this course. You will get most out of it when you have gone through that particular week's lecture, then come to the live sessions, and then ask your doubts. So this will actually help you understand the subject. It's a very, very, very translational subject, if I say, right? Animal physiology, it's a subject of sheer importance, obviously, because it is related to our physiology, understanding of ourselves, at least at a physical and physiological level, right? And what is the need of this course? I guess I don't need to say much for this particular thing, right? Because we know that we want to live forever, though we cannot. But if we cannot live forever, but we can live a quality life, correct? So diseases or illnesses, these create some hindrance in living our best life possible. From it varies from individual to individual, right? And there are other socioeconomic parameters which are associated with living good life, or at least whatever life you are living, it should be at its optimum and at its best, so that you are not suffering from any particular disease. Even if you are suffering from any particular disease, so the research community is working day and night to make sure that you that we can help, right? And I hope that this course and these live sessions will motivate some of you to get into research and put your valuable time and energy in enhancing the understanding of animal physiology and helping in curing illnesses, or at least mitigating the effects of certain illnesses or sufferings, right? It's a noble cause. The results are delayed, the gratification is delayed, but when it comes, it's like tremendous. Research field is very, very, very intriguing. So I hope some of you will join in research and specifically related to animal physiology. And as you might know, just give me a minute. Yeah, sorry for that. There was someone at my door. Yeah. So, as you know, that this particular week was dedicated to neuronal illnesses, right? Or say neurodegenerative illnesses. Even in the last week, I discussed about a particular disease called Alzheimer's. I hope you guys remember it. And if you haven't seen those videos have already been uploaded on my YouTube channel. You guys can go and check that out. So I've discussed about Alzheimer's disease. What are the known reasons which we are aware of that this is a particular reason cause of which Alzheimer's developed, right? Excuse me. So we have discussed those. And neurodegenerative disease in general is a huge challenge. Why? Because neurodegeneration, broadly speaking, we can categorize them into early onset and then age related, right? Neurodegeneration is oftenly associated with age related. So we are a population, we in general, like throughout the world, obviously from country to country, the proportion of young individual to old individual changes, but everyone will get old at some point, right? And these neurodegenerative diseases are progressive. Progressive meaning that they will start showing their symptoms as you age, correct? So, concerning thing about a neurodegenerative disease is that one of the bottleneck in neurodegeneration or identification of a neurodegenerative disease is that early onset, or say early detection, right? If it is an early onset disease, you can obviously check it. But if it is a age related degeneration or age related prognosis, then the bottleneck comes that how we can detect the disease before it has crossed that threshold that there is no coming back now. The individual has to suffer, correct? So early detection in any disease, whether it is neurodegenerative disease or a cancer or any, for example, CV, like cardiovascular diseases, right? Early detection is the key. Prevention is always better than the cure, right? So in any particular disease, the bottleneck always comes to detection, because factors we can control to some extent if we are aware of them. For example, we know that getting out in a direct sunlight may cause cancer, skin cancer, right? So what we can do is we can use some sunscreen, we can keep ourselves covered up, or if it is not needed, we should not move like directly into the sun, right? So these are certain things which an individual can take steps forward, can take certain steps towards protecting oneself. But if a disease is coming from the inside, it is not from any external source, then it is difficult to prevent it, right? Because we don't know what is going inside. So early detection is actually a key in controlling any disease. Though we might not have the cure for most of the diseases, which we're gonna see in the coming slide, but if we are able to detect it, we can mitigate the symptoms, we can prepare ourselves, and it won't be like a shock or a surprise, right? That, oh my God, after 50 or after 60 years of age, now the individual is showing a symptom of Alzheimer's. Now they have lived their most of their life, right? Average life expectancy is 75, 80 years, right? Again, depending upon from country to country, it varies. So so early detection of a disease is a key point here. And again, the same trend can be seen in the neurodegenerative diseases such as Alzheimer's or Parkinson's or ALS. People are working in making certain uh, assays or tools or different ways or techniques so that they can detect early in the life whether the person is predisposed towards having a particular disease. If it is a genetic disease, we can check that, right? The science has advanced this much that if it is a genetic disease which might show its symptom or which might show itself up, we will be able to see that in the genome itself. If we get the individual person sequenced their genome, we can tell them that you might get this disease, right, in later year of life or throughout your life. Then they can make certain lifestyle choices based on this information to make sure that either the disease should not present itself, because at the end of the day, if it is a genetic disease, chances of it showing up is higher. But if there are certain lifestyle measurements or certain lifestyle choices which an individual can make, which can help in delaying the onset of a disease, then obviously the person will go for it. Correct? But for diseases such as neurodegeneration, we still do not have exact markers. And what I mean by markers, or in this particular case, biomarkers are special kind of signature. Either it can be a protein signature, it can be a lipidic signature, or it can be uh, anything different from the normal, right? Which we can pinpoint and say, if this signature is coming in your profile, you are susceptible to a particular disease. And in this case, for example, we are talking about neurodegenerative diseases. Correct? So identifying biomarkers in itself is a huge research field. Because if you can identify markers of a particular disease, you can surely say that this individual might get this disease or is suffering from this disease, and then we can take our recommended steps or preventive steps. Correct? So most of the work for particular diseases, in this case neurodegenerative diseases, a section of researchers are actually working on knowing the biomarkers, right? So that as soon as even a small amount of that marker shows in your profile, we can warn the person and we can keep them under surveillance so that they should not suffer a lot, or we can make their diagnostic things or their treatment profile, all those things accordingly, because then we'll be having a lot of time to manage the disease. Correct? Once the disease has onset and it is progressing, it is hard to control. If we talk about cancer, right, so there are certain markers for different types of cancer, whether it's a solid cancer or a lymphoma or those things, they have particular biomarkers associated with them. With the help of biomarkers, we can actually say that the individual is susceptible for this disease, or they have a probability of getting this disease, right? Because there are certain cancers which occur due to mutation in the gene, right, which are heritable. So we can actually tell the person that you are susceptible for this particular type of cancer. It is not necessary that you might develop it, but you have a probability or inclination or more probability at the end of the day, more probability of getting this type of a cancer, right? Similarly, for neurodegenerative diseases, a lot of work has been done into it. And why such important section of scientists are working for this, we will understand this if we look at this distribution of the neurodegenerative diseases according to the uh, global location, right? According to the sections of the world. So you can clearly see that in most of the sections, the first cause, the primary cause or the primary disease, which leads to the maybe mortality or suffering. At the end of the day, these are for mortality, right? At the end of the day is stroke, which we commonly call as cardiovascular disease, right? Stroke is heart attack. Heart attack disease related to heart are called cardiovascular diseases. So stroke is the number one throughout the world. Even globally, it is the number one. Correct? Now, if you look at migraine, which most of the people suffer from, and you can look, and I guess that most of you know someone who suffers from migraine, right? So it is that common. You can see in the world globally, it is ranked second, which means people suffering from stroke is the one. Second is the migraine, and look at the third disease, what it is. Third disease is a neurodegenerative disease, which is Alzheimer's disease and other type of dementias. Correct? So Alzheimer's disease, which belongs to a neurodegenerative disease, is actually globally ranked third. Correct? And imagine in certain regions such as East Asia or Southeast Asia or Oceania, it is literally ranked second. So after cardiovascular disease, people belonging to these regions, they have Alzheimer's. Correct? And in the previous week, if you guys remember, that we actually understood what causes like what is the reason for Alzheimer's, right? It was due to the accumulation of some type of protein. We'll see that today also, but it was that we're gonna discuss about Alzheimer's today itself. Look at the fourth one, which is meningitis, epilepsy, spinal cord injuries. All these are related to what? They all are related to neuronal system, right? Except first rank, which is a CVD, that is a cardiovascular disease. All top four or top five, out of top five, top four belongs to neuronal system diseases, indicating that if we understand our neuronal system to a better extent, we might be able to cure or at least prevent or at least treat most of these diseases. And imagine the impact. For example, if you went into a lab and you were looking into for example, Alzheimer's disease, something, and you came up with a cure. Imagine how many people want to benefit from your research. Correct? It is literally the world's third number of disease. Correct? Similarly, if you look for other diseases which belong to neuronal system, at the end of the day, you might be helping a lot of people directly or indirectly. Whatever research is done in the neuronal field might not impact directly, but it will put some information and knowledge into the field, which gradually people will pick it up. And due to the like logarithmic growth of research, it will like exponentially increase. Your little input will exponentially increase, and at the end of the day, you never know that you actually have a sure shot cure for Alzheimer's or Parkinson's or ALS or other neurodegenerative diseases, right? That is the reason why we are discussing neuronal system to this extent, because you can clearly see that globally, if we look at, even like spinal cord injury is also associated with neuronal system, and but it is accidental, right? So in accident, most of the time, if things are damaged, they are your neuronal system only, because spinal cord is literally running through your half of the body, right? So any injury, the probability of your spinal cord getting injured is very high in any type of accident or in any type of injury if it is not related to muscular, right? So the chances of your neuronal system getting injured is probabilistically high. Correct? So these are the reasons which I wanted you to understand why we are putting so much effort and energy in understanding our neuronal system and understanding the disease associated with it, because globally there's a huge requirement and need for people who actively work in the research of neuronal system, because there are many people who are suffering from it, and it will gradually increase. In the coming slide, I'm going to show you a trend. In the coming year, it is actually going to increase because people will age, right? And it's a age related disease. Correct? So any one of you want to take a guess that why these diseases only show themselves at a later age or at a later stage of your life? Anyone want to take a guess? I'll give you 1 minute for it. I'll give you a hint. These particular neurodegenerative diseases are called progressive diseases, progressive neurodegenerative diseases. So try to take a hint out of it, and just try to guess that why these diseases are showing themselves up when we are aging, right? So I hope that you guys have the answer for it. At least you might have guessed. The answer is because these events, if we talk about particularly about, for example, Alzheimer's, right? So there was an accumulation of a protein. So during age, right? During age, the accumulation gets deposited. It started occurring at an early stage, correct? At an early stage. It didn't start at a later stage. No, it started at a very early stage, but due to non-clearance of the accumulations, and particularly talking about Alzheimer's, and due to non-clearance of it, with age, they get deposited and accumulated in the localized environment, which throughout your age or in the later age expresses themselves as the cause of the disease. Correct? So this is one reason. So with age, the disease doesn't progress dramatically. No, the disease started early, but due to the accumulation, it crosses the threshold because it's slowly depositing, right? Due to the non-clearance, and when it crosses that threshold of accumulation, then we see the symptoms. Correct? That is why even when things have started at a middle age, not at a older age, at a middle age, the symptoms show themselves in the later stage of your life. That is why it is called progressive neurodegenerative disease, because it is progressing. It is a continuous work. It is not a one night event. For example, today all of a protein suddenly got accumulated and you got Alzheimer's. No, it's a progressive disease. It is progressing throughout. Correct? So most of the things are like this in a progressive neurodegenerative disease. The classical example is obviously Alzheimer's. So if we look at a global trend, right now what we saw globally what are the diseases which are contributing to the illness or the mortality of people, right? So now we'll see in the neurodegenerative diseases what are the diseases which are contributing to the maximum, because there are multiple types of neurodegenerative disease, right? Few of them we have discussed. If you have went through the this week 7's lecture, you know there were three particular diseases which we have discussed. First one was Alzheimer's, then Parkinson's, and then ALS. Correct? So among those, if you can see this first pie chart, we can see that these from this distribution, we can clearly see that the top contributor of a neurodegenerative disease is Alzheimer's, right? And it is associated with dementia, obviously. And I have told you in the last week lecture that though it is not a painful disease, but it's a hurtful disease for the patient as well as for the people who are taking care of the patient, right? So Alzheimer's people might not suffer physically, but they suffer a lot mentally and emotionally. Correct? And socially, obviously, socially. Then second contributor in the neurodegenerative disease is Parkinson's disease. Correct? So 30% of the total neurodegenerative disease. This data is of, I guess, 2016. So the numbers might change in the current scenario, but you can get a hint out of it, right? So out of 100%, 30% people, 30% of the neurodegenerative diseases is actually Alzheimer disease. Then 18% is the Parkinson disease. Correct? 14% is the Huntington disease, which we have not discussed much about, but you can always go back to the sources and look for at least some basic information about these neurodegenerative diseases if you are interested. Correct? Then the amyotrophic lateral sclerosis, which is ALS, right? And next one is the ataxia. These are the top 5 neurodegenerative diseases, out of which we have discussed the top 2 ones. Correct? Looking at the tissue, obviously, we know that brain will be the one, because we are talking about neurodegenerative disease, right? So brain will be the one. So even in the brain, hippocampus is the one which is mostly getting affected, right? And in the coming slides, we'll discuss about the Alzheimer as well as Parkinson. And I hope that you guys know Alzheimer's affect hippocampus, Parkinson affect some different area of the brain, right? So they all are brain related diseases, but they affect different region of the brain, and hence their symptoms, their treatment, their approach is very different. In Alzheimer's, it is mostly dementia is the major symptom, right? In Parkinson, it is related to the motor activity, that you are not able to control your motor actions. You cannot sit still, because a particular part of brain which is affected in Parkinson is actually responsible for sending signals and receiving signals from outside to inside, and letting the individual know what to do, what is the strength you have to use. For example, if I need to pick this pencil, right, I'll pick it like this, correct? But I cannot pick a glass like this. I have to pick a glass like this, correct? So all these motor decisions based upon the stimuli and the surrounding is taken by the brain. If there is a lack in the communication from the outer environment to the internal environment, or from internal environment to the outer environment, obviously our motions will be altered. And this is a very clear-cut seen in the Parkinson disease, correct? So you guys can go through this particular graph closely. I'll move ahead with this one. I just wanted you guys to see this, that me talking about neurodegenerative disease, obviously, as I said, that in research, you need to work for it, right? And why you need to work for it? Because at the end of the day, everything comes to money and funding, correct? For every biological research, whether it is basic sciences or translational work or any associated work with R&D related to diseases or human physiology or animal physiology or anything related to any disease, at the end, what we want a treatment. Either that can be a full protocol of a treatment like CAR T cell therapy, or it can be a drug molecule, which is the most common form of delivering treatment. Correct? So if you are suffering from a headache, you will take a paracetamol or a Saradon, right? So those are small molecules which works at a receptor level or a cellular level, because someone has understood the mechanism why we got headache. And if we block this thing, or if we enhance something else, our headache will be removed. To do that, we need a small molecule or a drug compound, and that can be taken as a tablet, capsule, or whatever it is, correct? So similar thing is for the neurodegenerative disease. There can be two alternatives, either a drug or a small molecule, or a surgery. Surgery is the last resort, obviously, correct? So if we look at this report, which is for the current year and like which has predicted for the upcoming year, we can clearly see that the neurodegenerative disease market is anticipated to register a 7% during the forecast period. This is the enhanced increased percentage in the global market, from 43.7 billion US dollars in 21 to a valuation of 92 billion dollars US dollars, right, by 2032. The neurodegenerative disease market is anticipated to show promising growth prospects during the forecasted period due to the increasing frequency of neurological diseases, an aging population, and growing demand for Alzheimer's and Parkinson's disease treatment. Why we are focusing on only these two diseases? Because they are the top contributors in the neurodegenerative disease category. Correct? So you can clearly see that from 43.7 billion, which in itself is a very huge marketplace, right? From that, it is expected to grow almost 7% to 92 billion US dollars, indicating that there's a lot of scope for people to get into, do a real good research, either come up with a treatment or a mechanism or a protocol, or come up with small molecule drugs or any procedure, right, which can help in altering this trend, right, of increasing patients for Alzheimer's and Parkinson's. Correct? So I hope that this gives you some idea regarding like why we need to understand neuronal system to a deeper extent and take these diseases seriously. They take time. So one of the reason why the research is slow for neurodegenerative disease is that it's difficult to get the sample, right? And here our sample will be individuals who are suffering from it. And individuals who are suffering from it are generally older age people, right? So it is really, really difficult to get a good sample size or good sample for actually learning and understanding about this disease. Treatment is a secondary thing. We first should understand whatever we know about Alzheimer's or Parkinson's is not the 100% story. It is not the whole story. Those are the fragments of things which we have some proof that if this exists, it is a sure shot thing that it will lead to Alzheimer's or Parkinson or other neurodegenerative disease, right? We don't know the whole story. We don't know the source of anything. We don't know the point at which our system switches from normal to neurodegenerative disease, right? Until unless it is like genetic or something like that, we won't be knowing it. That at what moment our system switches from normal system to a disease system. Correct? So getting the samples or getting the individuals who are suffering from this particular disease it is difficult if you want to do invasive research, right? Or you want to do in vitro or particularly in vitro research, you need to take sample from it. So most of the work for neurodegenerative disease is being done in animal models, right? So in animal models, it can be either a primate model or a non-primate model. So for non-primate model, it will be rodents such as mice or right mice. So though they are conclusive, we have gained a lot by understanding the disease from animal models. Most of our work deals with animal model only because you cannot directly research on humans, right? You can, it is unethical. So even before using certain animals as a disease model or use them as for understanding a particular disease, you have to go through a whole lot of ethical committee, because even animals are living beings. They have their own identity, they have their own rights. So you cannot use animals just for the sake of research. So you have to be optimized about using them. So when you are getting into an research, and if any one of you gonna go into research, and especially for the like animal based research or animal model based research, there will be huge ethical responsibility associated with you, that you should not hurt the animal, you should not use them or misuse them, you should consider them as a living organism and you should respect them as well. So if your study needs, for example, 20 mice, right? So you have designed the experiment in such a way that it will require 20 mice models. At the end of the day, you have to remember that they are being sacrificed, right? Sacrificed in the name of science. So imagine if your research include 20 animal models, you want 20 samples from those 20 animal models, you need to sacrifice 20 animals, right? And it is a huge toll. So you need to be very optimized and very logical about approaching animal studies. So this is another bottleneck, because you cannot just randomly say, okay, I need 100 animal models because I'm gonna try and test every single thing. No, you need to do your work prior getting into an animal research, so that you get the only the amount of animals you need for that particular objective. You should not use hit and trial on animal model. You should be so thorough with the literature work that you know ki this amount of or this number of organisms will suffice my hypothesis. Okay. So if you want to understand more about neurodegenerative disorders, I have given a link for this particular review. It's a really good review. It gives you a very holistic understanding of most of the neurodegenerative diseases, especially the top 3 and top 4 ones, which we have seen in the previous slide, that Alzheimer's, Parkinson's, Huntington's, right, ataxia, ALS, all these things. So you'll get some idea that though they all are neurodegenerative diseases, but their occurrence, their site of occurrence, also their localization, their impact, their symptom, their treatment approach, their testing approach, their diagnostic approach, they all vary, right? Our neuronal system is so diverse that even just saying that you understand neuronal system is a huge deal, right? Because every single compartment of your brain is controlling a single aspect of your life, correct? And to understand and say that you understand all of your brain is a, it's a very like dumb thing to say, because you cannot until unless you have spent years and your lifetime in only studying brain or neuronal system, you can say that. And there are many people out there, I'm not criticizing or saying anything bad to anyone, because they have spent their lives in learning about brain, correct? And even then they themselves say that they don't understand it completely, right? Because it is so complex. Hence we need people to get into research and understand such a complex system to make it simpler, and hence helping the individual and the people to live a quality life, to live a better life, correct? So with this, I would like to now switch towards our question and answer session. In this particular session, as I have very less number of participants, I might end this soon. So I hope that you guys can see this video later on the YouTube. And if you have any question, you can drop me a mail. I have given my website link in the first page of the slide. You can always mail me. I'm approachable, and let me know if you have any confusion, right? So let's start with the first question today. So our first question is, what part of the brain is majorly affected by Alzheimer's disease? And I hope you guys remember. I'll give you 1 minute for this question. So your 1 minute starts now. Repeating myself again. If you know the correct answer, good for you. You should also know what other options mean. And if you don't know the answer, try to link it with the lectures which you have seen, and also the live session which you have attended, correct? Well, the correct answer is hippocampus. I hope you got the right answer. And if you don't remember the right answer, it's okay. No problem. You can go back to the lecture, then you can understand. So if you remember, the hippocampus is like this round structure, which we have discussed in the previous week. Also in the lectures, the sir has discussed it in a very nice way. So you can see that this is kind of a normal brain, not so normal because there is inflammation or there is preclinical Alzheimer's disease hints that there is onset of Alzheimer's disease. This is the mild to moderate one. As you can see, the area affecting the brain is gradually increasing, right? It is not localized anymore, correct? Then in the severe AD, you can clearly see that most of the brain is affected with this, leading to the dementia thing, right? That loss of memory and all the things which are associated with the Alzheimer's disease. And I hope that half of the animal physiology course is passed, and now you guys are okay in seeing some histological images or real cell images. So I did that on purpose since the beginning of the course, so that you guys will be okay in seeing the real images and understanding what actually looks like in the real life, right? So you can clearly see that these are the images from an Alzheimer's patient, and this is the area which is associated with the Alzheimer disease. And you can clearly see, if you remember, the Alzheimer disease is the reason of the Alzheimer disease, which we know of, is are twofold. First one is the accumulation of the amyloid, which causes the amyloid plaque, which you can clearly see in the second image, right? Here, you can see that there's a tense image. This is the plug. And also there was tau fiber, right? Tau fiber associated with the Alzheimer's disease. You can clearly see that this is the neurofibrillary tangle, which is due to the tau filament, right? And then there are granuloscular degeneration bodies. You can clearly see here, this leads to the like this accumulation and all those stuff related to Alzheimer's, correct? So if you guys are not familiar with this, I will highly recommend you to go and check out last week's lecture, because I, in the starting time itself, I have discussed about the Alzheimer's disease, and I have showed certain videos which were pretty self-explanatory, and you will get a good idea about Alzheimer's disease. Okay, great. So we'll go to our second question for the day. So our second question is, pyramidal neurons are, so pyramidal neurons are unipolar, bipolar, multipolar, or all of the above. So I'll give you guys 1 minute for this particular question. Kindly type your answers in the chat box. So the correct answer is multipolar, right? So pyramidal neurons are multipolar. polar neurons. What are multipolar neurons? So, multipolar neuron is a type of neuron that possesses a single axon and multiple dendrites and dendritic branches, correct? Allowing for the integration of a great deal of information from other neurons, correct? So these are the real images. You can clearly see that this is the major axon which is associated with this neuron, and you can see why it is called pyramidal, right? It's like this, a very dense cell body, and it looks like a pyramid, correct? And then there's one single axon, and there are multiple dendrites which are having further branches. You can clearly see in this confocal image this green filament. This is the pyramidal neuron, and pyramidal neuron, you can clearly see that they are multipolar in nature. So what are multipolar neurons? So multipolar neurons in general means that they have a single axon and many dendrites which further has branches, right? Dendritic branches, correct? So a multipolar neuron has multiple dendrites, and pyramidal neuron is a type of multipolar neuron. Okay, I hope that this is clear to you guys. And if you have any doubt, kindly text in the chat box. So our next question for today is, what human ability is majorly affected by Alzheimer's disease, right? I hope that you guys get this answer because we have discussed in detail about this particular disease. So what human ability is majorly affected by Alzheimer's disease? Is it motor control? Is it hearing? Is it sensory control? Or is it memory? So you have 1 minute to answer this question, and your time starts now. Ten seconds to go. Type your answer in the chat box. Yeah. So the correct answer is memory. Obviously, we have been talking about neurodegenerative disease and dementia simultaneously with Alzheimer's only. Correct? So it affects our memory. And from the previous question also, we know that it affects our hippocampus, and hippocampus is associated with our memory system. Correct? So how the disease progresses is just a very small recap to what we have discussed in the previous week. So again, suggesting you to please go back to previous week live session, or you can go to the lecture videos of professor, and he has also discussed about the Alzheimer's disease, correct? So if you remember, there was amyloid precursor protein, correct, which was associated with the cell membrane. So if it gets cut at a particular location, then it is susceptible to get cleared from the cell. But if there is certain changes in the cutting site or the enzyme which is cutting the protein, then the protein is not easily cleared from the surrounding. and which leads to the accumulation, right? So that was amyloid precursor protein. This APP is amyloid precursor protein. So first step is the amyloid precursor protein being there, and then it gets cut by various enzymes, and the name of the enzyme, type in the chat box if you remember it, good for you. If you don't, go back to the lectures and tell me the name of the enzyme. Yeah. So then the protein gets cut into fragments, and if the fragment is cut properly, it won't get accumulated. But if the fragment gets cut not at the right place, it leads to the accumulation of this amyloid precursor protein, which further leads to the formation of plaque and these dense bodies, which we have just seen in the previous slide, right? Those were dense areas in the histology of the brain area. They leads to the accumulation of these plaque, and they are called amyloid plaque. Correct? To understand this again in a better way, I have this small video which describes the progression of the disease in a really good manner. Okay, so let me share my screen and we'll see about it. Okay. 1 minute. Yeah.
All these new trends on the grid.
You have to tell me this also, that you guys are able to hear the video and listen to it. Okay, and I'm going to share it. I hope that it is visible. Okay, yeah, I guess you guys can see it, right? So, excuse for the
Chatty season, you gotta go full glam.
excuses for
Catty, doughy, and boxy eyes. But is that a trend for my sleepy eyes? So
so all I can do is mute it, right? So we're gonna see about the Alzheimer disease in detail. It's a really good video. So okay. So I hope you guys can see and hear it. Yeah.
Over a century ago, a German doctor called Alois Alzheimer spotted anomalies in brain sections from a patient with dementia. Ever since, people have been studying the strange plaques and tangles that he saw, in the hope that we could one day understand and cure what is now known as Alzheimer's disease. Plaques are insoluble deposits of a peptide called amyloid beta, or A-beta. They're formed when a protein called amyloid precursor protein is sequentially cleaved by two enzymes, beta and gamma secretase. Other molecules are generated by this cleavage and may play a role in the disease, but A-beta is the main culprit. A-beta tends to misfold and become sticky, eventually clumping together to form soluble oligomers. Some of these aggregate into large insoluble fibrils that deposit in the brain as plaques. The oligomers come in several forms or species. We don't know exactly which species is toxic, but research shows that they weaken communication and plasticity at synapses. This could be what stops the brain from forming or retrieving memories. Neurons aren't the only cells affected in Alzheimer's disease. Astrocytes and microglia also play a role. Microglia are immune cells that clear out waste and prune synapses during development. Microglia take up A-beta, but they also get activated by it, triggering the release of inflammatory cytokines that can damage neurons. The microglia also start to remove synapses by phagocytosis. As synapses start to malfunction and neurons die, abnormal patterns of activity emerge, and eventually the brain can't process and store information properly. Another key feature of Alzheimer's disease is neurodegeneration. Neuronal death and damage is triggered by A-beta, but some of A-beta's effects seem to be mediated by another protein seen in the brains of patients, tau, a component of tangles. In a healthy neuron, molecules are carried along the axon on a series of tracks, made of microtubules and stabilized by tau. But in Alzheimer's disease, tau is modified, causing it to dissociate from the microtubules, adopt an abnormal shape, and move from the axon to the cell body. Like A-beta, tau comes in a variety of forms, and we don't know which ones contribute to the disease. And like A-beta, these forms either remain soluble or stick together and deposit as the tangles that Dr. Alzheimer saw. Eventually, these processes kill the neuron. Another problem seen in animal models is that misfolded tau proteins can spread across synapses into healthy neurons. There, they make healthy tau proteins start to misfold as well, spreading pathology across the brain. The pattern of spreading through the different brain regions matches the changing symptoms from early to late stages of Alzheimer's disease. This pattern also reflects how certain neurons are more vulnerable than others to dying. Despite these advances in our understanding of Alzheimer's disease, no cure exists. While drugs are being developed to target amyloid beta or tau, it's unclear whether they will eventually be successful in treating the disease. There's only one certainty. Continued support for basic and clinical research will enable us one day to diagnose and treat this devastating condition.
Correct. So as we have seen in this particular disease that uh, the memory is altered, the neuron get degenerated due to the accumulation of plaques, and plaques is due to the APP protein getting accumulated in the brain, correct? So those are the certain reasons for which the disease present itself, but we don't have a sure shot cure for this, correct? We can only target those things which we are aware of, correct? And here we know that, okay, this is a particular enzyme which cuts this protein is the reason for the disease, so hence we are targeting these checkpoints of the progression of a disease, correct? But still we are not completely sure how Alzheimer disease present itself. There are still much more work needs to be done in this particular field to get to the ultimate cure for this particular disease. Okay, moving on to the next question. So what the next question states is that what human ability is majorly affected by Parkinson's disease? Okay, so they both were Alzheimer's as well as Parkinson are neurodegenerative disease, correct? So in this particular question, we are asking about Parkinson's disease. So what human ability is majorly affected by Parkinson's disease? Is it motor control, hearing, sensory control, or memory? So you have 1 minute to answer this question and drop your answer in the chat box. the correct answer is motor control. So if you remember in the lectures, we have actually discussed about the Parkinson disease as well, alongside with the Alzheimer's disease. So there's a different region of the brain which is affected in the Parkinson disease in comparison to the Alzheimer's disease. So if you look at the region which is affected in the Parkinson disease is the substantia nigra part of the brain, which is responsible for producing dopamine. And I hope that you guys are aware of dopamine. So dopamine is a neurotransmitter, right? It's one of the common neurotransmitter, which I guess most of us are aware of. So to even again, to understand these neuronal diseases, we should know what is the source, what is the cause, what is the reason, like for treatment purposes. So whatever we know about Parkinson's, again, same with other neurodegenerative disease, though this is the second most common neurodegenerative disease globally, we can globally we can say uh, that we still do not know much about it, and it's a very, very, very active area of research, correct? So we'll see a small video about Parkinson's. This will help you guys in understanding the disease and also the severity, as well as the trouble which the patient as well as the caregiver has to go through when someone suffers from Parkinson. Okay, so just give me a minute. I hope you guys can see my screen and hear the video. Yeah.
Parkinson's is the second most common neurodegenerative disease after Alzheimer's. The most visible features of this disease are a slowness of movement, muscle rigidity, and tremors at rest. There are also many, often debilitating non-motor symptoms, some of which may present before the motor problems. Parkinson's affects many areas of the nervous system and different types of neurons. However, much focus has been placed on neurons in brain regions associated with the overt motor symptoms, notably a region of the midbrain called the substantia nigra pars compacta. This region forms part of a major pathway in the brain that's critical for facilitating movement. In Parkinson's, dopaminergic neurons in the substantia nigra gradually die, leading to the malfunction of this pathway and the characteristic motor problems. Drugs that replace or mimic dopamine are often used to treat these motor deficits, but they grow less effective over time. Deep brain stimulation may also be used to treat symptoms. However, no current treatment slows the neurodegeneration. A distinctive pathology in most cases of Parkinson's is clumps of misfolded proteins within neurons. Lewy bodies are the most common type. A characteristic component of these is a misfolded protein called alpha-synuclein. These molecules can form small repeated units called oligomers, or longer fibrils. Mounting evidence indicates that these are toxic to neurons and play a key role in driving Parkinson's. Unwanted proteins are normally cleared by the cell's different types of protein-degrading machinery. Some evidence points to these systems being overwhelmed by misfolded alpha-synuclein, and this might be a key factor in what kills the neurons. Parkinson's has also been linked to problems with mitochondria. Mitochondria provide cells with the energy to perform vital functions. They're highly dynamic and can fuse together or break up into smaller versions in response to a cell's energy demands. They can also be transported to areas of a cell that need them the most. However, in Parkinson's, these processes can be impaired, and mitochondria are unable to sustain proper neuronal function. As they become old or damaged, mitochondria are removed and replaced. Again, this recycling is thought to be disrupted in Parkinson's, leading to the accumulation of damaged or worn-out mitochondria. Another idea is that the cells called glia surrounding neurons may play a role in Parkinson's. As dopamine neurons are lost, One particular type of glial cell, called microglia, is thought to take up the resulting cellular debris, triggering an immune response. Once activated, they release inflammatory cytokines, which activate neighboring microglia and another type of glial cell called astrocytes. Chemicals released by activated microglia and astrocytes have been shown to injure neurons. It remains unclear which mechanisms drive the disease process in Parkinson's. What is clear is that with time, more areas of the nervous system develop pathology. And one emerging idea is that this happens through the transmission of misfolded alpha-synuclein. While the multifactorial nature of Parkinson's disease progression might make it hard to fully understand, it does provide a number of potential therapeutic targets for slowing or even halting the neurodegeneration. For example, enhancing the clearance of abnormal proteins and blocking their transmission, improving the function of mitochondria, and targeting the neuroinflammation are all currently active areas of research.
Correct? Now you can understand, right, why we need to understand and study about these diseases, because the more we know, the more things we can target for particular diseases, correct? So in Parkinson, the motor control system is affected. In Alzheimer's, the memory function is affected. Okay. So our next question for today is, neuronal processes in what part start to die off in Parkinson's disease? We have just saw in the video, right? And also in the lectures, and also I have discussed earlier. So is it amygdala, substantial nigra, hippocampus, or preposterior complex? I'll give you guys 1 minute for this. And for this particular question, I would appreciate you guys if you know little bit information about all the options. Obviously, the correct answer I want, but also something about the other option you should be aware of. Okay. And your 1 minute starts now. Last 10 seconds. So the correct answer is substantia nigra, right? We have just seen in the previous video. And you can clearly see that it's a very small region, substantia nigra. And what neurons or things are affected in Parkinson? It was a dopamine neurons, right? Dopaminergic neurons. So if you remember from the lectures also, there is one particular treatment which is very effective against Parkinson's disease. And I guess we have a question related to this in the coming slide. So because of this knowledge and information that this area is rich in dopaminergic neuron, so if we have something which mimic dopamine, or which is related to dopamine in any way, might be a cure for Parkinson's disease or might be treatment for Parkinson's disease. Okay, so this is how research works. Okay, so moving to our next question. What is the question? The question states that okay, it is the same question. So what neurotransmitter was originally secreted by neurons before dying off in Parkinson's disease? And I hope every one of you answers this correctly. Also, you should know about other type of neurons also. Okay, that's our deal. We should know everything about something about most of the options which are presented in front of us. So, which neurotransmitter was originally secreted by neurons before dying off in Parkinson's disease? Is it GABA, glutamate, dopamine, or caffeine? So, your 1 minute for this particular question starts now. Though I know you guys don't need 1 minute for this question, but still, I have to consider everyone's need. And I guess 1 minute is more than enough for this particular question. I hope that most of you get this answer correct. Obviously, the correct answer is dopamine. We just saw in the previous slide itself that in substantia nigra, the dominant type of neurons which are present in this region are the dopaminergic neuron, and dopaminergic neurons are the ones which secrete dopamine, which is a neurotransmitter. Okay, so you can see in the disease condition, there is drastically reduction in the dopamine secretion, which leads to the this Parkinson's disease, the symptoms associated with the Parkinson's disease. Okay. So this is a very old paper. I guess it was published in 2019, I guess. But it's a very good overview, particularly for the Parkinson's disease. If you remember, in the beginning of today's lecture, I've given you a review article. So review article will cover most of the neurodegenerative diseases. But this particular article, this is solely dedicated for the Parkinson's disease. So I will suggest you guys to go through this particular paper. I have given the screenshot of it. You can directly search in the Google and get the paper. Just for the hints, what you can expect from this paper, I'll just read the small abstract and then you'll understand what to expect from the paper, particularly with respect to the Parkinson's disease. Okay. So in Parkinson's disease, we call this as PD now onwards. So PD is the most common neurodegenerative movement disorder. In Europe, prevalence and incidences rates of PD are estimated at approximately 108 to 257 per 100,000 and 11 to 19 per 100,000 per year. It's a very high rate, okay, respectively. Risk factors include age, male gender, and some environmental factors. The etiology of the disease in most patients is unknown, but different genetic causes have been identified. Although familial form of PD account for only 5 to 15% of cases, studies on these familial provided interesting insights on the genetics and the pathogenesis of the disease, allowing the identification of genes implicated in a pathogenesis and often critical insights into the mechanism of the disease. Imagine whatever information and knowledge we have about Parkinson is from the familial type, which means the genetic type, which is associated due to the changes in or alteration or mutation in the genetics. But that factor for the progression of for the disease is only comprised of 5 to 15%, meaning that 95% of the time the occurrence of the Parkinson, we are not aware of the why it is occurring in patient, right? So how we can treat it if we don't know the cause of the origin of particular disease or the reason why this disease is representing itself, then how you can approach for the treatment, correct? So that is why when I said that, especially for the neurodegenerative disease, whatever we know is just a spoonful of information. We don't know much about it, and we need more active research and more work has to be done, particularly for the neurodegenerative diseases. Okay, continuing, the cardinal motor symptoms of PD are tremor, rigidity, bradykinesis, akinesis, and postural instability. But the clinical picture includes other motor and non-motor symptoms. Okay, its diagnosis is principally clinical, although specific investigations can help the differential diagnosis from other form of Parkinsonism. Pathologically, PD is characterized by the loss of dopaminergic neurons in the pars compacta of the substantia nigra and the accumulation of misfolded alpha synuclein, which is a protein associated with the Parkinson disease, which is found in intracytoplasmic inclusions called Lewy bodies. Currently available treatments offer good control of motor symptoms, but do not modify the evolution of the available treatments. But do not modify the evolution of the disease. This article is intended to provide a comprehensive, general, and practical review of PD for the general neurologist. Okay, so if you actually even understand 5 to 10% of this whole article, you will have a pretty good understanding of what is Parkinson disease, what are the common factors which we are aware of are responsible for Parkinson disease, and how we are handling and tackling the disease. Okay, so do try to read this particular review article. Our next question for today is, which of the following is a treatment for which of the following is a treatment for Parkinson's disease? See, I have already given you a hint. Let's see. Yeah, so which of the following is a treatment for Parkinson's disease? First, is it L-GABA? Second, is it D-GABA? Third, is it L-DOPA? And last but not the least, is it D-DOPA? So I'll give you guys 1 minute for this particular question and your time starts now. Last 10 seconds to go. Kindly drop your answers in the chat box. Yeah. So the correct answer is L-DOPA. I hope you guys were able to give the correct answer. Uh, and if not, I will request you guys to go back to the lecture videos, because we have discussed a great deal about this treatment, because it was a game changer for Parkinson's patients. So it's a it was a big deal, it still is a big deal, but there have been more modifications and alterations of this approach. But when it came, this L-DOPA treatment, it was a game changer for Parkinson's patient. So what exactly is L-DOPA? So L-DOPA, also known as levodopa, and L3,4 dihydroxyphenylalanine, is an amino acid that is made and used as part of the normal biology of some plants, okay, and animals, including humans. Humans, as well as a portion of the other animals that utilizes L-DOPA, make it via biosynthesis from the amino acid L-tyrosine. L-DOPA is a precursor to a neurotransmitter, dopamine or norepinephrine, commonly also known as noradrenaline, and epinephrine, also known as adrenaline, which are collectively known as catecholamines. Further, L-DOPA itself mediates neurotrophic factors released by the brain and CNS, that is central nervous system. So what was lacking in Parkinson's disease? What was enriched in Parkinson's disease area, that is substantia nigra? It was dopaminergic receptors, right? Dopaminergic neurons, sorry. So there was issue with the neurons, and hence there was reduced level of dopamine. So with this information, what we did, we created a precursor for the dopamine itself, which will increase the dopamine concentration and hence alleviate the symptoms of Parkinson's disease. Okay, so it's a pretty straightforward approach. But at that time, even to get this idea, to have a precursor in hand and to have a precursor which can actually get transmitted through blood-brain barrier, it was a huge achievement. Okay, so this is a very basic pathway of how L-DOPA actually is giving rise to dopamine. You don't need to remember everything, but the things which you should remember, that tyrosine is the precursor of L-DOPA, and L-DOPA is the precursor of dopamine. And dopamine was the neurotransmitter which was reduced in a disease condition, that is in Parkinson's disease. Okay. So again, I have a small video related to the L-DOPA, so that you guys have better understanding of the treatment which is currently available for Parkinson's disease. Let me share my screen.
L-DOPA is an amino acid.
I hope you guys can hear, and yeah, great.
L-DOPA is an amino acid involved in the synthesis of the neurotransmitter dopamine. When dopamine is produced in the brain, the amino acid tyrosine is first converted to L-DOPA, and then L-DOPA is converted into dopamine. While dopamine administered as a drug cannot pass the blood-brain barrier to enter the brain, L-DOPA can, and this characteristic has helped to make L-DOPA the most common treatment for Parkinson's disease. Parkinson's disease is characterized by the death of dopamine neurons in a region of the brainstem called the substantia nigra, and a subsequent deficiency of dopamine in a group of structures known as the basal ganglia. This dopamine deficiency is thought to contribute to the problems Parkinson's patients experience with movement. L-DOPA has a remarkable ability to reduce the symptoms of Parkinson's disease in some patients. The generally accepted mechanism for this action is that the brain is able to use L-DOPA to synthesize more dopamine and replenish its depleted dopamine stores. While this does seem to be a main characteristic of L-DOPA's mechanism, however, it is thought that L-DOPA may exert a therapeutic effect through other mechanisms as well, such as by acting as a neurotransmitter on its own. While L-DOPA can effectively treat the symptoms of Parkinson's disease, it is not able to stop the neurodegeneration caused by the disease. Thus, Parkinson's disease continues to progress even with L-DOPA treatment. Additionally, over time, L-DOPA's effectiveness begins to decrease, and eventually movement problems, known as L-DOPA dyskinesias, may occur in response to L-DOPA treatment. L-DOPA dyskinesias typically involve quick, involuntary movements, but may also include a number of other complications like uncontrolled muscle contractions or writhing movements. The mechanisms underlying L-DOPA dyskinesias are still not completely understood, and may involve factors like the continued death of dopamine neurons in the substantia nigra and fluctuations in dopamine levels caused by L-DOPA treatment.
Hair fall shampoo. Right? So we can actually see how L-DOPA works, correct? At least to a to our basal understanding or basic understanding of how we have targeted a particular disease by knowing the associated area through which the disease is progressing. Okay. So our next question for today is, which of the following is not a glial cell? Is it astrocytes, oligodendrocytes, Schwann cells, or neurons? So I'll give you guys 1 minute for this particular question, and I hope that all of you get it right. Okay. Don't worry about the text written here. You will understand why I have mentioned it here later. Okay, just drop your answers in the chat box. Yeah, so the correct answer is neurons. And why is that? So you guys need to go back to the lectures and see the classification of the neuronal cells, that they were glial cells and non-glial cells, and glial cells are of like 3 different types depending upon their location as well as function. So they were astrocytes, oligodendrocytes, and Schwann cells. They belong to the glial cell population, while neurons do not belong to the glial cell population. Now this text, why this text was here, it was because of this particular image. So this is how these glial cells actually look like in real life, at least on the plate, right? So these are astrocytes. You can clearly see why they are called astrocytes, because they are spreading everywhere like a star, and they have multiple angles and multiple this outspread things, right? And then we have oligodendrocytes. You can clearly see that sorry, this is this is oligodendrocyte, and this is the representative way of how oligodendrocyte is represented. This is a node of reindeer. I hope you guys know what is a node of reindeer, and this is the myelin sheath. The green part is the myelin sheath, and this is the microglia cell. You can clearly see here how a microglia cell looks like. So both, so all three, astrocyte, oligodendrocyte, and microglia cells, these are glial cells. While neurons, they do not belong to glial cell category. Okay. So our next question for today is, programmed cell death is called. So it's a bit different from the core neuronal system. It's a generic microbiology term. So I hope that you guys are aware of it. And if not, don't worry about it. We'll discuss, even right here, we'll discuss. Also, in the lectures, if I remember correctly, not exactly it was discussed, but in the coming sessions, it will definitely be discussed for sure. And if even if you are not able to understand, and if in the lectures it was not cleared for you guys, you can always drop a mail, or you can drop your query even in the YouTube window. Okay, I'll respond to it. So programmed cell death is called apoptosis, necrosis, cell injury, or external chemical induced cell inflammation. So I'll give you guys 1 minute for this question as usual, and type your answers in the chat box. Last 10 seconds to answer. Yeah, so the correct answer is apoptosis, and I hope that you guys got it correctly. So what exactly is apoptosis? Apoptosis is the process of programmed cell death. It is used during early development to eliminate unwanted cells. For example, those between the fingers of a developing hand. In adults, apoptosis is used to get rid of the body of cells that have been damaged beyond repair. Apoptosis also plays a role in preventing cancer. If apoptosis is for some reason prevented, it can lead to uncontrolled cell division and the subsequent development of a tumor, right? So we need balancing of both things. We need cell division also, and we need clearing of the cells also, right? So if any of the mechanism, whether the cell is dividing uncontrollably, or whether the clearance mechanism through which the cells are getting cleared, or because they are old now they need to be cleared, they need to be dead, right? So if any of the mechanism is altered, it leads to the very common disease, that is cancer, correct? If both the operations are opposite, for example, cell division is uncontrolled and the apoptosis is decreased, it will lead to cancer, correct? Because on one hand the number of cells are increasing, also this supports this, the apoptosis part is also not getting the cells cleared or not letting the cells die, which ultimately leading to the increased number of cells, and those things are called tumors, and tumors later, if they metastasize, then they are termed as cancer. Okay. So what happens, it's a very simplistic approach to understand apoptosis. But for example, if a cell is there and its time has come, the cell needs to die. So how this programmed cell death or apoptosis occurs is that there will be a condensation, there will be internal signals from the cell. And again, there are multiple molecular small molecules which are associated, biomolecules which are associated, which will tell the cell that it is time for you to die now. And this process is called apoptosis. So there will be chromatin condensation, which is one of the major steps to for the cell to be cleared from the system. So there will be chromatin condensation, then all the material will be get autophagocyte. Autophagocytosis basically means that the cell will be ruptured into small balls, small parts, and later it will be cleared by the macrophages. Okay, then these apoptotic bodies formed, and then this is cleared by the macrophages, which are always roaming here and there and expecting a thing, because they just like to eat everything and anything. Okay, but to understand more about apoptosis, and you guys should know that both apoptosis and necrosis is a cell death phenomena. In both of these processes, cell is being killed and is cleared out from the system. But there is a huge difference between apoptosis and necrosis. So we will try to understand this difference by looking a small video which clearly distinguishes between necrosis and apoptosis. So I hope that you guys will be able to hear it and see it and understand it. Okay, so here it goes.
Necrosis versus apoptosis. Hello everyone. This tutorial will explain to you the main difference between apoptosis and necrosis. Introduction. All cells in a multicellular organism undergo growth and death. Cellular death is essential for an organism to grow and survive. Human body is made up of innumerable types of cells in various parts of the body. There are mainly two types in which a cell undergoes death, either through exposure to harmful environment, injury, or through a pre-planned and regulated process of disintegration. Cell death occurs via two different processes, one being necrosis and the other being apoptosis. Now the question is, what is necrosis? Necrosis is a process of cellular death, which occurs when cells are exposed to extreme conditions, which are very different from normal living conditions. This leads to damage to the internal cellular environment and rapid cell and tissue damage. So the basic inability of the cell to maintain balance when exposed to rough conditions leads to complete discord in the cell's functioning and eventually its death. What is apoptosis? Apoptosis is a pre-planned regular process of cell death occurring in the body where the cell itself actively takes part in its own death. It occurs as part of normal cellular multiplication and turnover, so cells die naturally to maintain a balance of the cellular multiplication. Apoptosis is required for smooth functioning of the body. Most cells in the body have a shelf life. For example, red blood cells live for 120 days. At the end, they are destroyed in the body. This is a method of apoptosis. Difference in cause and presentation. Necrosis is a pathological process which is detrimental to the body. It occurs when the cells are exposed to toxins or on exposure to extreme conditions, which could be anything from increased temperature, decreased oxygen levels, etc. These conditions lead to damage to the wall or membrane of the cell. An inability to maintain normal functioning in these conditions leads to an imbalance in the internal environment of the cell. This leads to inflammation and tissue damage, which leads to accumulation of cellular debris. Apoptosis is a normal, natural process which is necessary for the body as it maintains a balance in the number of cells in the body. If the cells do not die on time, it can lead to a tumor or cancer formation, which is an accumulation of unwanted cells. If cells die too fast, it can lead to conditions such as AIDS, heart disease, and liver disease. So if apoptosis does not take place as it should, there will be signs of disease. Difference in structural changes. In necrosis, there is a loss of integrity of the wall of the cell, which leads to swelling of the cell's contents as well as disintegration of small bodies of the cell. In apoptosis, there is no disintegration of the cell membrane or wall, rather there is a shrinkage of cellular contents along with clumping of the membrane. This leads to formation of condensed bodies known as apoptotic bodies. These bodies are recognized by the immune system of the body and destroyed efficiently by
I'm, uh, I'm extremely sorry, but I guess the video got ended. I don't know why this happened, but it's okay. We'll start from where we have left. We are at the end of this discussion about the difference between necrosis and apoptosis. So I'm continuing.
due to which there is no accompanying inflammatory reaction. Difference in biochemical reactions. Necrosis is a passive process which does not require energy and can occur at any point of time. This is a random event which is unregulated. Apoptosis is an active process which requires energy and occurs in an organized manner. Many enzymes and agents are required to carry out various steps of this process. Summary. Apoptosis and necrosis are variants of cell death that vary greatly in all ways except the end result. Necrosis is a type of cell death wherein the cell dies an untimely death due to uncontrolled external factors, and apoptosis is a predefined cell suicide wherein the cell actively destroys itself to maintain smooth functioning of the body. Apoptosis is a natural physiological process which is required by the body, while necrosis is a pathological process which stems from inflammation and tissue damage.
So I hope that you guys can now have a basic understanding of the difference between necrosis and apoptosis, as both of them are cell death mechanism, but one is programmed and the other one is spontaneous. Bringing my question that I don't know whether you guys are aware of this or not, but their sunburns are very common in foreign countries, right? Those people who go for sunbathing, so if they are not using proper sunscreen or proper shade, or if they are left or if they are in out sun for a longer duration of time, they actually get sunburned, right? So according to you guys, what will be the sunburn leads to? Like, what is the underlying reason why sunburn happens? Will it be apoptosis or will it be necrosis? So this is a question for you guys to figure it out for the next week. Okay. Yeah. So for last question for this particular session, the jumping of nerve impulse from one node of reindeer to another is called orthodormic conduction, antidormic conduction, saltatory conduction, or action potential. Your 1 minute for this particular question starts now. So the correct answer is saltatory conduction. So a saltatory conduction is the rapid method by which nerve impulses move down a myelinated axon with excitation occurring only at the nodes of Ranvier. So nodes of Ranvier are the places where there is no myelin sheath. Okay. And if you have attended the lectures, you know that these areas are oftenly very much densely packed with sodium channels, okay, especially voltage gated sodium channels. If you remember from the action potential graph, voltage gated sodium channels are the ones which are responsible for the depolarization of the neuron, or say action generation of action potential. To see how this saltatory conduction actually happens, okay, it's a very brief, very less descriptive um, video. So we'll quickly go through it. Yeah, and I hope that you guys can see that. There's a ad. I cannot
Meet Jay, a neat.
So yeah, I'm sorry about that. Yeah.
Conduction through myelinated nerve fiber. Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction, and it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membranes following a series of action potentials being propagated along the fiber.
Right? So I hope that you have some idea about the saltatory conduction, and even if you still have certain doubts, I would suggest you to go back to the lectures. There we have discussed the structure as well as the saltatory conduction itself. And if you have any doubt, as again I say, I'm always available via mail. You can directly drop me a mail, and also you can drop the comments on the YouTube if you want to, and I'll try to respond to them. Okay, so for today, this is all from my side for this particular week. I hope you guys learned something new. And again, I would like to congratulate you all for like almost completing half of this course, and I hope you guys are learning and understanding new things. So as you know, I post question to ponder upon. So for this week, it will be what are the differences in physical and chemical properties of blood and cerebrospinal fluid that flows in the blood-brain barrier. And if you haven't heard this blood-brain barrier term or anything, these things were discussed in the lectures. So please go back to the lectures and try to complete the lectures in that week itself. So a quick revision of what we did today. So we understood about why neuronal system is important because it's prevalent. What are the major contributors of neurodegenerative disease? What type of disease contributing majorly for this particular section? And what is the projected market value for neurodegenerative diseases? Then we discussed about our regular questions which were pertaining towards Alzheimer's, then pyramidal neurons, multipolar neurons, then Alzheimer's disease reason, then again somewhat related to their cause, their why they are presenting themselves in older age, and all those basic discussions. Then about Parkinson's disease, that what area is affected, what type of neurons or neurotransmitters which are affected in the Parkinson's disease. And with this information, how we can actually help in treating Parkinson's disease, right? By knowing this information only, then we can do that. Then we understood a very basic classification of glial cells, that how do they actually look like under the microscope, as well as in their representative way. Then we understood a very different topic, which is not solely associated with the neuronal system, but it is a generic molecular topic about apoptosis. We understood about the difference between apoptosis and necrosis. I've given you guys one question about sunburn, so you need to tell me the sunburn is a apoptotic phenomena or a necrotic phenomena. Then at the end, we understood why nodes of Ranvier are required and how they are associated with saltatory conduction. And this will make you understand that this is how we are so quick in learning and doing things, right? Because we are conserving energy. We are not traveling throughout the neuron. We are traveling through only to the nodes of Ranvier. Okay, so this is all from my side. I hope you learned something new, and thank you so much for your patience. Thank you.